So thank you for the introduction. I'm just showing you my industrial declarations and saying that I have no conflict of interest. So as I mentioned in the last talk, I do research in the Blizzard Institute, and I'm based within the Centre for Cell Biology and Cutaneous Research. What I'm going to talk to you about today is a very severe disorder called Harlequin ichthyosis, and how we have developed a 3D model, which we think reflects many features of the disease in skin. And we've used that model to try out treatments which we think may be helpful for patients. So this is a patient with Harlequin ichthyosis at birth. So you can see there's this massive hyperkeratosis of the skin with fissuring. Quite often there's deformity of the fingers and toes. There's very marked ectropion of the eyelids and eclabion of the mouth. Generally, the ears are very bound down, but after several weeks, this thick layer of skin sheds, and you're left with a very red person. So this is an adult with Harlequin ichthyosis. She's about 30 now, and you can see that her skin is very inflamed. She still has a deformity of her fingers. The severity of the inflammation causes hair loss and sometimes altered pigmentation. And when you look at adult Harlequin ichthyosis skin, you can see that the epidermis is acamphotic or thickened, and there's also a very compact thickened stratum corneum compared with a normal basket weave stratum corneum. So the Harlequin ichthyosis gene is called ABCA12, and it's an ABC transporter, so it's a transmembrane protein. And ABCA12 seems to be particularly important in transporting glucosal ceramide from the lamellar bodies into the lamellae of the stratum corneum. And basically in skin, you've got two types of fats, polar and neutral. So cholesterol, ceramides, and free fatty acids are examples of neutral. And glucosal ceramide, sphingomyelin are examples of polar. This is an electron microscopy view of Harlequin ichthyosis skin, comparing normal skin on the right with Harlequin ichthyosis skin on the left. And these are the lamellar bodies. And you can see that in normal skin, you get these little pouches, which are basically transporting the glucosal ceramide and other good things into the stratum corneum. And these are absent in Harlequin ichthyosis skin. So mutations in ABCA12 can cause Harlequin ichthyosis, i.e. the phenotype that I've just shown you. But they can also cause very severe congenital ichthyosiform erythroderma, stroke lamellar ichthyosis. In black, you can see the Harlequin ichthyosis mutations, which occur throughout the gene. These can be missense, nonsense, or splice site mutations. You can also get insertions shown in blue or deletions shown in red. Deletion phenotype is generally quite severe. And in green, grey, purple, and orange, you see mutations causing other types of ichthyosis, i.e. the phenotype, but they all have severe ichthyosis. So we did RNA sequencing on Harlequin ichthyosis patient skin. We got biopsies from 4 adults, and we compared them to 5 normal age-matched controls. We initially had over 1,500 hits, 824 downregulated and 714 upregulated genes with quite stringent stats. So we had a p-value of less than 0.01 and a fold change of greater than 2. So we did functional annotation clustering using David and Reactome. And as might be expected, we found genes involved in epidermal differentiation, for example, keratins. We found genes involved in regulation of epidermal lipids, for example, ALOX12B, genes involved in desquamation, such as kallikrein-1, and genes involved in inflammation, in particular the IL-1-like cytokines, IL-36A and IL-36G, and genes involved in interferon gamma-mediated response, such as STAT1. We did some staining on Harlequin ichthyosis skin, comparing it with normal, and we found that STAT1 was slightly upregulated, particularly in the granular layer of Harlequin ichthyosis skin. We then looked at phospho-STAT1. We saw no phospho-STAT1 phosphorylation in the epidermis of normal skin. However, in Harlequin ichthyosis skin, we observed nuclear phospho-STAT1 in all 3 samples. HI1 we observed less, probably because that was in the freezer for 18 months by the time we stained it. So these antibodies work better in fairly fresh samples. So the JAKISTAT pathway, as you all know, is a major pathway in cells involved in response to cytokines. SOCS genes basically inhibit JAK, and these genes are involved in cell cycle arrest and apoptosis or cell death. They're involved in immunosurveillance. They're involved in inhibition of angiogenesis or blood vessel formation. And they're also involved in nitric oxide synthesis. So there's a gene called INOS, which is important in generating nitric oxide from L-arginine. So INOS seems to be important in inflammation as well as many other things. And we observed increased expression of INOS in Harlequin ichthyosis epidermis. We also observed increased expression of IL-36 alpha and IL-36 gamma, interestingly within the nucleus of keratinocytes as well as cytoplasmic in Harlequin ichthyosis skin. IL-37 is an inhibitor of the IL-36 family of cytokines, and this seemed to be reduced in Harlequin ichthyosis skin. We then went on to use a technique called CRISPR-Cas9 gene editing, where Cas9 enzyme and a guide RNA attaches to matching genomic DNA. So you have to design a guide RNA to match the genomic DNA. This Cas9 guide RNA complex cuts the DNA. If you wanted to do gene therapy, you could make pre-designed DNA to go into that cut, but that wasn't our plan. What we wanted to do was make a deletion. If you go to this link here, there's a very nice video which explains this technique. So for our in vitro Harlequin ichthyosis model, we made a CRISPR-Cas9 ABCA12 knockout cell line. We used a human telomerase immortalized keratinocyte. So our wild type cell line was the control, and the knockout cell line was the cell line for the patient model. So we picked exon 27 because this seems to be an important area in the transmembrane protein. And we made this deletion here. So here you can see the electropherogram of the wild type, the ABCA12, versus the mutant. And these are the details of the mutation. So using qPCR, we checked ABCA12 mRNA expression, and it was markedly reduced in the mutant cell line. Using Western blotting, we checked protein expression. We were unable to detect protein expression in the knockout cell line. We looked at the behavior of the cells in 2D. And here you can see that the ABCA12 knockout cells looked a bit bigger. And this is a quantification of the normalized cell area, so you can see it's increased. And growth seemed to be a little bit slower. So this is a growth curve over 5 days, and you can see that the knockout is a bit less than the control. We then went on to make our 3D organotypic model. To do this, we used these inserts. We poured collagen and matrigel matrix into the inserts with fibroblasts. We then put keratinocytes on top and allowed them to settle under the media. Then the media was moved, so it's just covering the bottom of the well. And you've got your fibroblasts in a now contracted matrix with keratinocytes on top. And then over time, the keratinocytes will stratify and you will get an epidermis over a 2-week period. So when we did H&E staining on the 3D model, we saw that the ABCA12 knockout model was different. The epidermis was thickened. There was a very early onset of differentiation, and you got this abnormal stratification in the upper part of the epidermis, whereas the wild type epidermis had a fairly nice compact stratum corneum. In adult patient skin, which I showed you in the first picture, you can see that we've got the same thickened epidermis. We don't have the same thickened stratum corneum, but I think this area here sort of represents the abnormal stratum corneum. In Harlequin ichthyosis skin, you see early onset of the marker involucrin, which is normally expressed in the stratum granulosum of the epidermis. And in the knockout model, we also showed increased expression of involucrin and early expression compared with control. So as I mentioned, polar and neutral fats are a very important part of the skin barrier. And you can visualize these with Nile red staining. So cholesterol, for example, will stain green, and glucosamide ceramides will stain red. So here you can see that in adult patient skin, in Harlequin ichthyosis, staining is gone. In the 3D organotypic model, we've got the exact same finding. So you've got a nice barrier in the wild type, and it's gone with ABC knockout. So the Harlequin ichthyosis knockout mouse is neonatally lethal, and it's neonatally lethal because the skin barrier defect is so severe. So the mouse will lose protein and water through the epidermis and basically will shrink and die. And using a toluidine blue permeability assay, you can see that the mouse stains blue, whereas the control barely stains at all. So we can sort of imitate this in the organotypic model using a lucifer yellow dye test. It's actually green. So basically, in a functional barrier, the dye is retained at the top of the epidermis, whereas if the barrier is impaired, the dye will go through the epidermis. And here you can see quite nicely in the Harlequin ichthyosis model, the dye goes through the epidermis. In the wild type control, the dye is retained on top. So to look at the faithfulness of the model compared with Harlequin ichthyosis skin, we also used RNA sequencing of the 3D model. And we saw changes in genes, but not as many as in the Harlequin ichthyosis skin in the 3D model. So the top panel here is the groups of genes that were altered in the skin. And the lower panel is the group of genes that were altered in the 3D model. Red is upregulated and black is down. And here you can see you've got keratinization, metabolism, immunity, and interferon gamma in the skin. And basically in the 3D model, we don't have interferon gamma signaling, probably because there were no immune cells. In Harlequin ichthyosis skin, this is just a string diagram showing some of the genes of interest that are altered. And these include STAT1 and STAT3, IL-23A, IL-17, IL-36A, IL-36G, IL-37 was down, NOS2, and IL-1A and IL-1B. IL-36 gamma was massively upregulated. It was 900-fold upregulated in Harlequin ichthyosis skin. IL-17 was less, it was like 5-fold or something like that. So, as I mentioned before, SOCS are inhibitors of JAKs, and we found that these were downregulated in the Harlequin ichthyosis 2D model. So here you've got SOCS1 mRNA expression and SOCS3 mRNA expression, and both are reduced in the Harlequin knockout model. Because there are no immune cells in the model, we didn't expect to see any phospho-STAT1 expression in the knockout epidermis, and that was the case. So we saw no expression whatsoever of phospho-STAT1. However, when we took the cells and exposed them to interferon gamma, we did see increased phosphorylation of phospho-STAT1 in the knockout model, which we've quantified here. We also looked at INOS in the model, and we saw very nice upregulation of INOS in the knockout model, which we've quantified here. We also looked at the NO intracellular concentration in an assay, and again, this was markedly upregulated in the ABCA12 knockout model. We used this INOS inhibitor, 1400W, which is a potent and selective inhibitor of inducible nitric oxide synthase. Here you can see in 3D we've treated cells with the inhibitor and then looked at the NO intracellular concentration. You can see that the inhibitor reduces the increased NO concentration back towards normal. We then put this into our 3D model, and here using H&E, you can see that there's an improvement in the model with the INOS inhibitor. So you've got the knockout here and the treated knockout below it, and you can see you get some nice regeneration of the stratum corneum, which is quite like the control. We also saw improvement in barrier permeability. So here you can see that in the Harlequin ichthyosis model, the barrier is totally permeable, but it seems to be improved by the INOS inhibitor back towards the control level. And we also showed an improvement in Nile red staining, suggesting that an intact barrier is forming in the ABCA12 knockout model after treatment with the INOS inhibitor. And finally, quite specifically, when we looked at glucosal ceramide staining, you can see that this is most marked in the upper epidermis in the control, and it's lost to a certain extent in the ABCA12 knockout. And when we treated the 3D organotypics with the INOS inhibitor, this was restored. And just to remind you, ABCA12 is important in transport of glucosal ceramide by the lamellar bodies into the lamellae at the upper part of the epidermis. So we then had a look at the JAK inhibitor Tofacitinib, because STAT1 and STAT3 both came up as possible targets in Harlequin ichthyosis skin from the RNA sequencing and our staining of Harlequin ichthyosis epidermis. So here you can see the control on the left and the Harlequin ichthyosis model on the right. And you can see that there's a very convincing restoration of staining at the upper part of the epidermis, indicating that there is now a barrier in the Harlequin ichthyosis model after treatment with the Tofacitinib. And you also get the feeling that there's some restoration of the stratum corneum, and the epidermis isn't quite as thick as it was. But I have to say, I'm not totally convinced by that, but the barrier certainly is extremely convincing. We think this is an off-target effect because we could not detect phosphorylation of JAK1, JAK2, or JAK3 in Harlequin ichthyosis epidermis in the 3D model. It has been shown in both eczema and psoriasis that Tofacitinib increases expression of filaggrin, but I think increasing expression of filaggrin alone would not be enough to cause this, improvement. So in summary, we've identified hits from RNA sequencing, and I've taken INOS as an example here. We've followed up candidate genes and validated these. Here you see increased INOS staining in Harlequin ichthyosis skin. And then we've treated with an INOS inhibitor and shown that we can restore the skin barrier. So if you want to read more about this, we published this paper in the JCI around July of this year. And this is the graphic from the paper. So you can see the JAK inhibitor and NOS2 inhibitor seem to restore a functional skin barrier. And what does that mean for patients? So current treatments include moisturizers, keratolytic, manual exfoliation. Some patients take acetretin, but this is unsuitable for females of childbearing age because it's teratogenic. There are some ongoing trials in ARCI. So there's a trial ongoing in the US of secukinumab, which is an IL-17 inhibitor. And there is a trial starting of an IL-36 receptor inhibitor by a small company in the US starting soon. So INOS inhibitors and JAK inhibitors are also potential new treatments for severe ichthyosis. So I would like to acknowledge my research group, particularly Florence, who was the PhD student who worked on this project, and Matt Cayley, who helped her a lot. Priya also started some of this work, and initially she's not in the picture. David Kelsall, with whom I've worked on Harlequin for some time. The Harlequin ichthyosis patients who contributed to the study, and Anton Inright, who helped us with the analysis of the RNA sequencing. I would like to also acknowledge GSK and the BBSRC for funding, and the MRC. So thank you very much.